DNA divides in a unique way. First, the double helix strand of DNA gets winded by an enzyme called helicase. During winding it unzips that section of the DNA, in the sense that it breaks the hydrogen bonds between the nitrogen bases. Another enzyme called the RNA polymerase is introduced to the leading strand of the DNA, the one on the left. The RNA polymerase reads the first few nucleotides of the leading strand and gives them their corresponding bases, adenine to thymine, and cytosine to guanine. After this, the DNA polymerase reads those few bases, then finishes up what the RNA polymerase starts. It needs an impetus to begin its work. The ligand strand, replication is different, since DNA polymerase can only add nucleotides in the 5' to 3' direction. The RNA polymerase and the DNA polymerase must work in fragments to complete the task. First, the RNA polymerase reads a few bases, and moves on to another fragment, skipping a few. These few are filled by the DNA polymerase. This goes on until the entire strand is completed. Later, an enzyme called the DNA polymerase III extends the RNA primer with short lengths of complementary DNA. The DNA polymerase I digests RNA primers and replaces it with DNA. DNA as a ligase then joins the neighboring fragments together into longer strands. Those two new strands of DNA that split up, there ends the replication. Transcription is the first part of protein synthesis. In this process, the helicase, again, is involved in unwinding and unzipping of the DNA. This forms two strands, the coding strand on the left and the non-coding strand, the intron. The coding strand is read by the RNA polymerase, which binds to the coding strand at the top box. The initiation stage of transcription. It then creates a new individual strand of messenger RNA that is an exact copy of the coding strand. The only difference is that thymine is replaced with uracil in RNA. That is done by bringing free nucleotides in the nucleus and attaching them in their appropriate positions. This is called the elongation stage of transcription. Once the RNA polymerase reaches a terminal signal, it stops coding. The newly formed mRNA then undergoes splicing, where introns are preserved and exons are cut. A 5' tail is then added to the 5' end of the mRNA and a poly A tail to the 3' end. The mRNA is then ready to be sent out of the nucleus to undergo translation. The newly formed mRNA then goes to a ribosome consisting of RNA and proteins, with two binding sites in the ribosome, the A site and the P site. The mRNA strand is divided into triplets called codons, which will determine the amino acid sequence. This is the initiation stage of translation. The mRNA is in scanned in red. The complementary nitrogen bases of the triplets are bound by tRNA. On the bottom of the bases, on the top is the amino acid that corresponds to the anticodon. The anticodons are then bound to the binding site. While this happens, the mRNA keeps moving through at the ribosome while more tRNA brings in other bases amino acids. When a new tRNA is introduced, the first one moves over. When both binding sites are loaded and a new tRNA must be introduced, then the first tRNA transfers its amino acids on to the next tRNA.